This has been an exceptional week. It actually started, gosh, not too long ago, maybe 2 months. We had a conference call, and Blake Watson from GMO-free Hawaii Island said, wow, wouldn't it be wonderful if we had Tyrone Hayes come to Hawaii? That would be fantastic because, we've got these, 5 of the big 6 chemical corporations. We've got this unique situation where we're seeing combinations of chemicals being sprayed. That's something that's very unique to GMO technology. When I first started, looking at the OSHA spray tags in fields, I'd see atrazine sprayed, Roundup was written in, and maybe, Lorsban, all on the same tag. So these combinations are something never been studied, we contacted Dr. Hayes just a little over, a month and a half ago, and he said he would be glad to come to Hawaii. And I asked him, are you okay if, we'd really like to get you to every island. Would you be okay if we flew and spoke on the same days? And he said, load me up. So I've had the honor and the privilege to meet Dr. Hayes on Maui, meeting him for the first time, and we've been traveling all week long and speaking to filmmakers. The word is getting out that Hawaii has a story to tell, and people are coming here, and you guys are all helping to tell the story, and that's what's going to help. It's really hard for us to save ourselves from here, but as our story goes worldwide, we're going to get the attention. Nobody wants to see Hawaii ruined, and everybody across the globe is chanting, "Stop poisoning paradise." So you are going to enjoy Tyrone Hayes' presentation very, very much. It's so interesting. Dr. Hayes is a professor at UC Berkeley, a tenured professor. He has been studying atrazine in water and the effects on frogs. And we're going to learn a lot tonight. You're going to have fun, and you're going to leave well-educated and inspired. So I'd like to have you give a warm, warm welcome to our special guest, Dr. Tyrone Hayes.
Wow. Thank you, I'm honored. People here keep telling me thank you for coming. Thank you for coming. I agreed to come. I had to be here. You didn't. So thank you for coming, and thank you for your support, and thank you for standing, for all of us, for standing together for what we know is right. Before I talk tonight and tell you about my interesting title, From Silent Spring to Silent Night, A Tale of Toads and Men, I want to start with this phrase, with this proverb that I learned by working in Southern Africa. It's a Xhosa proverb from the people there. And loosely translated, it means people are people through other people. And it's always been a tradition of mine ever since even when I was a graduate student. Before I speak, before I tell you who I am and what I do, I thank the people who make me who I am, who are responsible for me being here. I always start with my family. This is an old picture, but my mom, my dad, my brothers, my sisters, my wife, my kids, my family is everything to me. I would be nothing without these people in my life. So thank you for their love and support. And certainly none of it would be possible. I'm a biologist. None of it would be possible without my parents, my father, my mother, Romeo and Susie Hayes, for their love and support over these last 47 years, especially the first early years. This beautiful woman, my wife, Catherine Kim, I would not have finished college if it wasn't for her love and support. Family have to love you. They're stuck with you. She had a choice, and she chose me, and I'm forever thankful for that. I want to thank my son and my daughter. They're all grown up now, 21 and 18. These are old prom pictures. And I have to tell this story. I don't know which made me prouder as a father, that my son borrowed my tie for his prom, or that my daughter borrowed my earrings for her prom. She waited 18 years of me telling her, no, Daddy's earrings are too grown up for you, sweetheart. And she finally got to go in my jewelry box. So to my daughter. I also want to thank my funding sources. A lot of people have supported the work that I've done over the last 20 years. It takes a great deal of money to do what I do. And this is also a matter of disclosure for me. I have been funded by Novartis, Syngenta, the makers of atrazine. In fact, they introduced me to atrazine. They don't like to talk to it. Well, let's just say this. There are a lot of Syngenta executives that regret that they listened to Jessica Badu and called Tyrone. I also want to thank the students that have been involved. Most importantly, everybody in blue is an undergraduate, and a special person that's there in green is a former undergraduate of mine. She's now a graduate student. She's there in green. She's the first Native Hawaiian to get a PhD in integrative biology in the history of the University of California. She's from Ilo, and she's actually here visiting now. I got to see her yesterday. I'm very proud of the work that she's done. And finally, I want to thank and actually dedicate this to my grandmother who passed away in 2005. But lucky for me, she passed on her love of education and her desire to make the world a better place through education. She passed that on to me before she passed on. She also taught me something very important. She taught me that if you want people to listen, if you want people to learn something, don't teach. Don't give sermons. Don't give talks. Don't give lectures. Don't preach. Just tell a good story. And so that's what I hope that I'm going to do today, is just tell you a good story. One of the exciting things for me about being a teacher is that you never have to stop being a student. And sometimes the things you learn are much more important than the things you come to teach. You might recognize that person in the corner there. Jerry, I want to thank, special thanks for bringing me out and for chauffeuring me around. I hope I haven't been too difficult. Feeding me. Two things I haven't been is bored and hungry. I can tell you that. I want to thank this person. This is from my first trip to Waimea a little over a year ago. And there I learned some important things in this visit. I learned one, I'm going to talk to you about the details of how pesticides affect molecules. I'm going to talk to you about the molecular level and what's going on. But there I learned some much more bigger things that you can see. I learned, for example, in the town of Waimea and the school there that's covered in dust that we know is laden with pesticides. You can see that there's a problem. You can see that there's a problem when you can go on Google Earth, and see the topsoil that you're losing here from your island that's flowing off into the ocean. Soil that's laden with pesticides. You don't need me to tell you there's a problem. You can see the problem from space. I learned in my visit to Molokai about a much bigger problem than what these big seed, big ag companies are causing on the islands. So, for example, I learned about this sustainable way of harvesting fish, where you build these ponds, and it requires, though, that there's a freshwater spring flowing off into the ocean to bring the fish in. But as these big seed companies draw the water table down, these springs disappear. And it's destroying an important part of culture and history and an important way of life. Not to mention that as they drop the water table, the brackish water will start to flow in, and also now all the water is mixed with pesticides that are going to impact the fish. You don't need me here to see that. You can see that happening now. I'm here to tell you about something smaller that I think will have an equal, if not bigger, impact as well, just to add to the arsenal of information that we need to stop this from happening. A few years ago, I'd never heard of atrazine. In fact, I read something today that sort of pissed me off a little bit. It said that I was, one, counting on atrazine for income. Let me tell you right now, I have never benefited financially, personally, from atrazine, except for the manufacturer who used to pay me $1,000 a day. I'd be making a lot more money if I'd stayed with them. The other thing it said, not to risk my modesty, but it said that I'm becoming famous because of atrazine. How many people have heard of atrazine before you heard of me? I'm not becoming famous because of atrazine. Atrazine's becoming famous because of me. Here's what it looks like if you care. It's a so-called S-chlorotriazine. It's an herbicide or a weed killer. It's mostly used on corn, but as many of you know, it's also used on sugar cane. It's used on golf courses. It's used in the tree farming industry. It's been used since 1958, and we use 80 million pounds in the United States. When I started working on it, it was the #1 selling pesticide in the world. It's now #2, but not because we use less, but because we use more of something else, and that's glyphosate. It's used in more than 80 countries, but it's now outlawed in all of Europe. And if you've heard me give this talk before, or if you've gone on the internet, you know that this slide is a lie. I'm feeding you a lie. The company keeps writing letters to me. Atrazine has not been outlawed in all of Europe. It has been denied regulatory approval by the European Union. I don't know what the difference is, but I know that this one pisses them off. So this is the phrase that I keep up there. Because that's just the kind of brother I am. That's what I do. So the company came to me and they said, we want you to study the African clawed frogs. So I'm going to tell you how I got into this whole thing with frogs. Then I'm going to talk to you about people and what's going on with people. And then I'm going to talk to you a little bit about the company and how well you can trust them. So they came to me, Syngenta. They were Novartis then. I'll tell you about their name change later. And they said, we want you to study the African clawed frog and find out if atrazine disrupts their hormones, if it's an endocrine disruptor of any sort. And now, for those of you who don't know, everybody uses the African clawed frog. Who's ever heard of it? It's a really well-known animal model for developmental biology. And it's well-known for a reason, I'm going to tell you, and it's four important reasons I want to tell you. It's used in biology because somebody in 1920 discovered that the human pregnancy hormone will make this frog lay eggs. So by 1940, this is a true story, by 1940, this frog was the pregnancy test. If you thought you were pregnant, you'd go to the doctor, they would inject some urine into the frog, and if the frog laid eggs, you were either happy or sad, depending on your situation. Now, here are the reasons that this story is important. One is it shows you the value of curiosity-based research. See, I'm just a little boy who likes frogs. I didn't go into this to do anything that was important that anybody would care about. But it shows you the value of curiosity-based research. Somebody in 1920, somebody sat down and said, hey, you know, I wonder what will happen if I inject pee into a frog. Like, how do you come up with that idea, right? But you can see how important it turned out to be. The second reason I tell you this story is because it shows you how similar we are to these guys. The testosterone that's important in our development, the estrogen, the thyroid hormone, are exactly the same as the hormones that this frog and all vertebrate animals use when they're developing. The human pregnancy hormone that's responsible for all of us, and it will be responsible for your children, is so similar to this frog's hormone that it will make this frog lay eggs. So as I tell you what atrazine does to this frog's hormones, you should be thinking, wow, this is a tale of toads and men. What might this compound be doing to me? The fourth reason that this story is important is they developed new pregnancy tests, and people then just threw the frogs out, these African clawed frogs. So now I can go to San Diego and Los Angeles and collect them for free and use them in my experiments. In fact, technically then that makes my frogs African-American clawed frogs, but that's a technicality that's irrelevant to the story I'd like to tell you. So while working for the company, I was working at the university, but the company was paying for part of my research. We first discovered that if you raise African clawed frogs in this atrazine at low levels, I'll tell you what the levels are later, their voice box, their larynx didn't grow properly. Now that's bad news because a male frog sings and a female doesn't for the same reason that men have deeper voices than women, testosterone. So these data implied that these frogs exposed to atrazine didn't have enough testosterone. So in hypothesizing that they didn't have enough testosterone, we decided to look at the gonad. There's going to be a lot of gonads in my talk tonight. I hope you don't mind. But that's where it all starts. So if you look at the gonads of some of these exposed animals, sometimes you get animals that have two testes, which is pretty good. Every guy should have a pair. But then this individual, you can see, not only has the 2 testes, but it's got 2 ovaries. It's got another large testis in case it needs more help. It's got more ovaries. Like, you don't even have to leave home. There's a whole party going on in there, which is not normal. And here's what I mean. This is not a judgment call. By not normal, I mean that frogs are not naturally hermaphroditic. There are male individuals and female individuals. There are fish, including reef fish off the coast of Hawaii, that are naturally hermaphroditic, but there are no frogs that are naturally hermaphroditic. So we knew that we had discovered something that presented a problem. Now, we hypothesized that the following was happening. If this big yellow circle is a testicle, the testicles should make testosterone. In fact, the word testosterone means testicular hormone. It's 2 words stuck together. We proposed that atrazine turned on aromatase. And I want you to remember, if you don't know the word aromatase, I want you to remember it. Aromatase is the enzyme or the machinery, if you will, that converts testosterone into estrogen. And the word estrogen means generator of estrus. It's the female hormone, which is fine if you're a female, maybe not so much if you're a male. The consequences are that your testosterone's going away, you're using it up, if you will, so you're demasculinized, but you're also feminized because now you're making the female hormone when you're not supposed to. So we tested that, we tested part of it at least, and we measured blood levels of testosterone in control of unexposed males. And when you compare that to atrazine-treated males and females, you see that these atrazine-treated males have much, much lower testosterone. They're not different from a female. So at this stage, since I'm telling you a story, at this stage I was coming up for tenure. I was a young professor. Novartis had said goodbye. Syngenta had said goodbye. But we published a paper, Hermaphroditic Demasculinized Frogs After Exposure to the Herbicide Atrazine at Low Ecologically Relevant Doses. And we published it in the Proceedings of the National Academy of Sciences. Who knows PNAS? It's a big deal. It's one of the top 3 journals in the world, one of the top 3 toughest journals to get stuff published in. So it was peer-reviewed and published in a really tough journal. In fact, I have to tell the story. My mom's going to kill me. But I called my mom, and I was just coming up for tenure. I said, Mom, we have a paper coming out in PNAS. First thing my mom says, she drops the phone. I said, did you hear me? We have a paper coming out in PNAS. My mom says, I heard you. How do you get a paper cut on your penis? I said, no. Completely foreign. She had no idea what I was talking about. She calls me back the next week. And she says, how important is that paper? This is a true story. I said, what do you mean, Mom? She said, I went to Barnes & Noble, and they never heard of that magazine. I tell you the story for a couple reasons. We're going to come back to that. But most of the stuff we do in the ivory tower means nothing to 99.9% of the world. And I'll tell you why that's important in a minute. So as important as I should also point out, 4 black men and a Latina co-authored this paper, which is also probably a record for the National Academy of Sciences. They didn't even know. A couple other things were important. As important as the paper was for me and my career and for Syngenta, some questions were left unanswered. For example, we didn't know if these hermaphrodites were males with ovaries or females with testes, because frogs don't have visible sex chromosomes. All we knew is that, for example, if we treated a group of animals, we might get 50% females, 30% males, 20% hermaphrodites. So we had a good idea that the males were being feminized, not the other way around. But we also didn't know what happens when they become adults, because it takes 4 or 5 years for them to completely grow up. And we only, we kind of basically looked at the little kids right after they metamorphose. So it took some years, but that's okay. I was in no hurry. I had tenure. So it took some years, but here's the answer. It turns out that by the time the paper came out, somebody had discovered a gene. You can see the bar at the top there. Somebody had discovered a gene that only females have. So we could tell who was a male and who was a female genetically. And so this guy who looks like he's smiling, he's a male. And he's hurt his leg, see? And that's his brother giving him a ride home. It turns out that like 10% of the exposed males completely turn into females. So, for example, you could see him there. He lays eggs and everything. In fact, he's a grandmother now. It's not something you hear every day. So I could have published that. But again, I was in no hurry. I had tenure. So we decided to look at what happens to the other 90% of the genetic males that don't turn into females. Are they competitive? And so we did these experiments that I call the pool party experiments. We literally just put a bunch of animals in a pool and see who gets the female and who doesn't. And if you do that, the atrazine-treated males almost never get the female. They can't compete. Only 2 atrazine-treated males ever in the presence of other males got the female. And if you look at their hormone levels, okay, you find out that the atrazine-treated males, on average, have much lower testosterone levels. But what's more important is you find out that the animals who actually make the love connection, shown by the hearts, have the higher testosterone levels. So these atrazine-treated males, in red here, don't have enough testosterone to be competitive. Either the females don't like them or they get beat up by the males with the high testosterone, we don't know. But they lose and they have lower testosterone levels. So then we did another set of experiments, which I actually call the Motel 6 experiments. In this case, we just got them a room. No competition, we just put males with females. And if you do that, we did a test where we let them have their night of, froggy style. And then we just counted how many eggs the male fertilized. And if you do that, here's what it looks like. Each female lays about 2,000 eggs. And if you do that, in the top left corner, that's an egg that's not fertilized. And those other figures are just various stages of development. What you find out if you do that, so it's a complicated test. There's an undergraduate sitting there going, 1, 2, 3, 4. But it's important. If you do that, you find out that control males fertilize about 85% of the eggs, whereas atrazine-treated males have a fertility rate that's down at 15%, for 2 reasons. One is they don't even try. You watch them, they just sit there and watch the females lay eggs. They don't even bust a move, as we say back home. Even if they do, I always ask, because there's students in the audience, you guys like p-values, right? You got to have p-values when you publish in the highfalutin journals. What I like better than p-values is when you can see the difference. Can you see the difference between a control and an atrazine-treated testis? So this is a sliced testis under the microscope. And what you're looking at, I'm going to blow this section up, is these are testicular tubules full of sperm. See, these are solid in the middle. With these atrazine-treated animals, I'm going to blow that section up. These are testicular tubules that are virtually empty with a little bit of cellular debris. So they don't have enough testosterone to show male behavior, even if there's no competition, and they don't have enough testosterone to maintain sperm production. So even if they show the behavior, they're shooting blanks, so to speak. So we thought we were done then. We published another paper in PNAS. Atrazine induces complete feminization and chemical castration in male African clawed frogs. The company hates the word chemical castration. That's why I put it in the title. Because that's the kind of brother I am. What's equally important there is that 7 undergraduates co-authored this paper, and Sherry Gallipo, my student from Hilo, was also a co-author on it, and that was very important to me. The next thing we set out is we wanted to know, does this effect only occur in this one species of frog, or is it a generic problem that we need to worry about? So we looked at North American leopard frogs, a very common frog in North America. If you went to school in the US, you probably dissected them. And it turns out if you treat these guys with atrazine, 0.1 parts per billion, you find that these are testes at the top, and all that stuff at the bottom, I call it the junk in the trunk, those are all eggs that are bursting through this male's testis that's been exposed to atrazine. Now, at this stage, you might read stuff on the internet that says, I wouldn't share my data, the company likes to write things like that. One, they own the data on the first species. It was not possible for me to not share them, the data. And 2, when the EPA, when I was doing this work, the EPA was in my lab reanalyzing the data and watching us do everything we did. They saw it. In fact, I sent an email, I still have it, to Tom Steger, the EPA. I sent them this photograph, and it broke protocol, right? Because I published this in Nature. I actually broke protocol. You're not supposed to share the data before you publish it. But I sent it to the EPA because I thought it was kind of urgent, right? And Tom Steger of the EPA emailed me back, and he said, thank you, Dr. Hayes. This is a very interesting finding. He said, however, we at the EPA don't find that it's an adverse effect that would trigger reassessment and regulation of the chemical, right? Now, here's the part of the talk where I'm a little bit sexist, because I only want to talk to the fellas for a second, right? My wife tells me that there is nothing more painful in life than childbirth. And I would guess, based on the G.I. Joe Kung Fu grip she had on my hand when my son's big head came out of there, I'm going to give that to her. But fellas, are you with me when I say that a dozen chicken eggs in my testicle would probably be somewhere in the top 5, right? EPA says, no, that's not an adverse effect. Keep spraying. So we kept spraying, not us. And what this study allowed us to do, this laboratory study, is it allowed us to go into the field and ask, is this a laboratory artifact, or is this really affecting frogs in the real world? So we did a study. I'll show you how it went in a minute. But we first asked, what are the levels we're talking about? Because you've heard about some levels. I'm going to talk about that in a minute. You've heard about the levels the EPA says is okay. So for reference, we're using 0.1 parts per billion. That's 0.1 micrograms per liter. That's 100 nanograms per liter. That's 100 picograms per mil. That mean anything to you? It don't mean nothing to me. So I'm going to give you a visual for what you can't visualize. That's about 1/1000th of a grain of salt in 2 liters. But hormones are active at levels that are like 100 times lower than that. So it sounds like a little bit, but it's enough. In fact, how relevant is that to the real world? Well, it turns out the package of atrazine recommends application at 2.9 to 29 million parts per billion. So a farmer could be using this stuff at levels that are 290 million times higher than we use in the lab. And if you go through the published literature, here's the minimum and maximum detected in agricultural runoff, temporary pools, and that's a log scale, by the way, permanent water, precipitation. Here's what we're using in the lab. So here's all the habitats that would be at risk. There's enough atrazine in rainwater, up to 100 times atrazine in the rainwater than we're using in the lab. 500,000 pounds of atrazine come down in rainwater every year in the United States. It can travel 1,000 miles. The U.S. Geological Survey says they can measure atrazine in the rain in Minnesota from when farmers are applying it in Kansas. That's why the European Union banned it. France banned it, Germany was still using it, and it would end up in the rainwater in France from Germany. To give you another perspective, that's what the EPA says is okay in your drinking water. 30 times higher than we know to be biologically effective. They never tested 3 parts per billion. They got cancer in rats at 3,000 parts per billion, and they divided by 10 because it was a rat. They divided by 10 because they only used 1 strain, and just for safety, they divided by another 10. They never tested 3 parts per billion, and they never looked at endocrine disrupting activities of atrazine when they made that decision. 30 times higher is allowed in your drinking water. So we did this study, and here's kidneys, there's a testis, and I'm going to show you what this looks like under the microscope. So that's what's called a transverse serial cross-section. But just imagine I'm slicing a salami. I fold out a slice. The color's different because of the stain, the dye that we use under the microscope. Now, if I blow that up, and now I blow that section up, what you're looking at now are testicular tubules, just like I showed you before, except instead of sperm, what's in here are eggs or oocytes. In fact, we published this paper in Nature, and I invented the word testicular oocyte. For some reason, it pisses them off when I make up words. They said, that's not a word. I said, yeah, it is. I graduated from Harvard. It's published. It's a word now. And I kept using it. Yeah, and you know why. It's just the kind of brother I am. So they're testicular oocytes. And so we did this study. That was from an animal from the wild, not my lab. And so we did a study where we drove across the country. And I usually tell some obnoxious joke about we control for latitude. But the reality is, if you follow those circles, I don't know if you can see that arrow. That's Highway I-80, and we were driving to a meeting in Indiana, and we collected frogs on the way and got a Nature paper. That's fuel efficiency right there. So it turns out, and the red areas show where there's most atrazine. My red dots are showing areas where there's atrazine contamination, as measured by a government lab. And every place we found atrazine, we found hermaphrodites and vice versa. But the reason that we got the paper published in Nature, another highfalutin journal, is not because of this correlation, but because we could take animals from a red dot and raise those eggs up in clean water, and they wouldn't come out as hermaphrodites. Or we could take animals from a blue dot, a clean site, and put them in atrazine in the lab, and they'd come out as hermaphrodites. So we could show that it just wasn't natural variation or coincidence. We could show cause and effect. That's why the paper got into big journal. So the next, and kind of the last little piece I'll tell you about frogs is, we wanted to ask, in a landscape like this is Nebraska, how important is atrazine? Because they're using a lot of other pesticides. And then later you'll see in this last study that's unpublished, we wanted to ask, how important are pesticides given all the other stressors that a frog or a population has to face in the real world? And so we did a study. We went to, this is a cornfield in York County, Nebraska, and we had a list of all the pesticides that were applied. In Nebraska, you have to post what's been applied, so you can just drive up to the field and see. And so there are tadpoles in this water, and just coming off this one field, they're exposed to all those herbicides in red, not just atrazine, the fungicides there in blue, and the insecticides in purple. And that's just off of that one field. And so we tested each one of these compounds individually or in combination. You see why there's so many undergraduates now. The experiment was all replicated. We raised 3,000 tadpoles individually, the whole thing. And I won't bore you with all the details. We published it in a journal that's from the National Institutes of Health. And we showed the following. Your natural stress hormones in people and in frogs, if you're under long-term stress, can do some bad things. It can cause immunosuppression, so you get more diseases. It can cause decreased growth, retarded development, and for frogs, inhibit metamorphosis. We showed that pesticides act as a stressor and actually increase stress hormone levels. And every time you add a new pesticide, it's like a new stressor. So for every new pesticide, you get more stress hormone that produces these effects. So now we started to see that the problems weren't so simple. Because if we weren't doing this in the lab, we'd say, oh, frogs are dying because they're getting diseases. Well, the reason they're getting diseases is because their immune system doesn't work. The reason their immune system doesn't work is because they have too much stress hormone. The reason they have too much stress hormone is because they're exposed to pesticides. But we could break all that down in the lab. Now we wanted to test this in the real world. So it's one thing to do it in the lab. And these data aren't published yet, so this hasn't been peer-reviewed or published. This is the Salinas River. It flows south to north, and it takes about 2 hours to drive the whole river. So that's about 120 miles. And the north, at the end of the river, there's a lot of agriculture. In fact, I always ask this question when I'm outside of California. Who's ever eaten anything from Salinas? Yeah, everybody raise your hand. I guarantee you, 85% of the country's lettuce comes from Salinas. 85% of the country's lettuce comes from about 60-mile stretch of Salinas. Incredible intense agriculture. So in a sense, we had an experiment where we can go down at the mouth of the river. There's a lake here and a dam, and there's no agriculture. This is where rich people in Monterey and San Luis Obispo get their water from. So we can go down to a clean area and examine frogs down there. Further down in Atascadero, there's no agriculture, but high stress, because the water's all been drained off for the ag further downriver. So, for example, that's about 3,000 tadpoles in an inch of water that's at 100 degrees. So they got incredible environmental stress. Further downstream, the water's back, 2.5 feet deep, 22 degrees, but 100% of that water is agricultural runoff. 100% of that water is water that runs off of your food. 100% of that water is water that's running off of a field that has a big sign on it that says, poison, do not enter. And somehow 2 hours later, in the grocery store, it's okay for us to buy it and eat it. So compliments of Google Earth, what we did is we looked at immune function, growth, and sex problems in animals from the protected watershed in Santa Margarita, animals that are stressed but not yet experiencing pesticides, and animals that have no other apparent environmental stress, but 100% of their water is agricultural runoff. And I won't bore you with all the data and all the graphs. A picture's worth 1,000 words. Here's the bottom line. These are representative tadpoles, and it's an invasive species, by the way. It's bullfrogs. But the only difference between this tadpole and this tadpole is that this tadpole is living in water that runs off of your food. They're in the same river. They're at the same temperature. They're the same age, the same species, the same developmental stage. They were collected about 2 hours apart. The only difference is that little tadpole with retarded growth is living in water that runs off of your food, our food. Here's where that spot is, where we collect in Salinas. And now I want to take a minute, because I had this fancy Harvard liberal arts education where they made us take these breadth requirements. We couldn't just do science. That's all I wanted to do. And we had to read books like East of Eden by Steinbeck. He also wrote of Mice and Men, not to be confused with of Toads and Men. But he wrote in 1952, he wrote this book, East of Eden, and I was flipping through it one day, and I came across the following passage. In 1952, Steinbeck wrote, Salinas was surrounded and penetrated with swamps and tool-filled ponds, and every pond spawned thousands of frogs. With the evening, the air was so full of their song that it was a kind of roaring silence. It was a veil, a background, and his sudden disappearance, as after a clap of thunder, was a shocking thing. It is possible if in the night the frog song should have stopped, everyone in Salinas would have awakened. It is possible if in the night the frog song should have stopped, everyone in Salinas would have awakened. Let me read that again. It is possible if in the night the frog song should have stopped, everyone in Salinas would have awakened feeling that there was a great noise. In their millions, the frog song seemed to have a beat and a cadence. And perhaps it is the ear's function to do this, just as it is the eye's business to make stars twinkle. Here's what Salinas sounds like now. I haven't heard a single native frog call in Salinas in the 6 years that we've been working there. And what I think is great about that passage is it's not from a scientist. It wasn't a scientist who went out and said, I want to estimate the frog population in Salinas. It was a literary artist. The frog song was such a part of that landscape that a literary artist spent a half a page talking about it. If the frog song should have stopped, everyone would have awakened. Well, the frog song has stopped, but most of us are still asleep. Nobody has awakened. I'm not going to tell you that it's atrazine that's taking the frogs out. I'm not going to tell you that it's pesticides. I could tell you the number one cause, habitat loss. But when you drain off all the water for agriculture, and the only place that's left for frogs to lay their eggs is chemically contaminated water, you've got a problem. When you throw into there now bullfrogs, that's a species that we've looked at, an invasive species that competes with the frogs, that's a problem. When those invasive species bring in diseases that the frogs aren't used to, and their immune system has already declined because they're in pesticide-laden water that's caused their stress hormones to go too high. On top of that, throw in climate change, and now temperatures is another stressor. There's no way possible that the native frogs will survive. The frog song has stopped, but we haven't awakened. Now, this slide is a slide from Lake Nabugabo in Uganda, where I've done some work, and I bring this up to illustrate the following. The runoff from that crop, which goes into these containers, is the sole source of cooking, drinking, bathing water for the nearby village there in Nabugabo. And I used to use this slide to show the link between environmental health and public health. Now I think there's no such thing. There's no link. How is there a difference between environmental health and public health? How do we get to the point in society where we think that somehow we're separate from the environment? That if the environment isn't healthy, we can still be healthy. There's not a link. They're one and the same. But somehow in my village, I just live here, my water, though, just comes from that watershed behind us. And we make these assumptions that because there's not, because I'm not going to get cholera or diarrhea from my water, we make assumptions that we have clean water, that we have nothing to worry about. I don't believe that that's true. I don't believe that that's true for me in Oakland. I don't believe that that's true for you here. I don't believe that that's true for anybody in the United States for the following reasons. I call this From Silent Spring to Silent Night as a play on Rachel Carson's Silent Spring. Rachel Carson wrote in 1962 that the death of birds in our pending silent spring should be a warning to us. And here we are, many, many years since 1962, over 50 years, and now we have silent night, which I believe pesticides have a great deal to do with the decline in amphibians. Over 70%, coqui aside, over 70% of the world's amphibians are in decline. A group of animals that survived the mass extinction that took out the dinosaurs. A group of animals that have been on this earth since the dinosaurs. 70% are now facing declines. A colleague of mine wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. What I've talked to you about in my work is not just correlation. We've done controlled experiments, and we haven't just done it on one population. We've done multiple populations, multiple species, multiple genera. The frogs that I look at aren't even in the same family. But if that doesn't convince you that there's strong evidence for causation, then this will, I hope. People have shown that the same problems occur in fish, birds and reptiles, and mammals, including laboratory rats and correlational studies in humans. So every vertebrate class that's been examined shows similar problems, unless you work for Syngenta. So you might hear this, Hayes' work won't repeat it. Yeah, the people who work for Syngenta claim they couldn't repeat my work. 12 papers have been published on amphibians showing the effects of atrazine, but it's not just amphibians. So I told you that what atrazine does was decrease testosterone and increase estrogen. There's evidence from around the world, not just from my lab and not just from frogs. These are just a few of the scientists who have shown negative adverse effects of atrazine. Several of us published a paper recently. There are 22 co-authors from 12 countries on this paper that have all independently showed effects of atrazine around the world on different animals. In fact, I invented the word gonadotoxin in this paper. Pissed him off. But it's a word now. It's published. Here's what we showed. I told you that atrazine causes testosterone to go down and decreases sperm production in frogs. Here's testosterone in salmon. You give him atrazine, testosterone goes away. This is a guy in England. I don't even know the guy. Independently showed this. Here's my frog data. This is rat data that was produced by the manufacturer. You give him atrazine, testosterone goes down. So it happens across vertebrates. We published the following figure in our paper. Here's my frog testis, right? On the left, sperm in the testis, give it atrazine, no sperm. This is a guy in Belgium who studied fish. Give him atrazine, there's no sperm in the testis. I don't know this guy. I've met him now, but I hadn't met him before this, independently. This is a guy in Argentina. I didn't know this guy. I've met him now. This is an alligator, a caiman, a big reptile. Sperm in the testis, give it atrazine, no sperm. These data were produced in Nigeria and Croatia. This is a rat with sperm in the testicular tubule. Give it atrazine, it's empty, there's no sperm. And this is a guy in Pakistan who showed the same thing in birds and quail. Sperm in the testis, give it atrazine, there's no sperm in the testicular tubules. So independent scientists from around the world on any animal you pick have shown that this is a problem, independently. What's more is, if you look at humans, this is Shauna Swan. You know, I should point out, as I usually do, I'm the only academic who brags about what I didn't do. Everything I'm going to talk about for the rest of my talk is what other people have done that have shown the same kinds of effects that I've shown you in my work. My colleague Shauna Swan showed in 2003 that if you look at men in Columbia, Missouri, men who have atrazine in their urine, 0.1 parts per billion, have a low sperm count and can't get their wives pregnant. I don't know what that means. That's just correlational, right? But if you have enough atrazine in your urine, 0.1 parts per billion, to chemically castrate a frog or fish, then you can't get your wife pregnant. It's just correlation. But in every lab study, in amphibians and fish and birds and rats, reptiles, we see that atrazine makes your sperm go away. Now, watch this. I spend a lot of time on airplanes, so I make all my PowerPoints move. This is a study. This isn't my study, done in 1993. Here's atrazine levels in field workers. Now punch that down. Here's atrazine levels in men who apply atrazine, 2,400 parts per billion. So men who apply atrazine have 24,000 times the atrazine in their urine than we know is associated with low sperm count in men in Columbia, Missouri. Men who apply atrazine have 24,000 times the atrazine in their urine than we use to chemically castrate and feminize frogs in my lab. One of these guys could pee in a bucket. I could dilute the atrazine in their urine 24,000 times, and I could use the atrazine in their urine to chemically castrate and feminize 24,000 buckets of 30 tadpoles each. I'm just a little boy who likes frogs, but this is what first got me using terms like environmental justice or environmental racism, because we know nothing about the reproductive health of these men. They're 90% Mexican-American. In many cases, these migrant farm workers have life expectancies of 50. And in addition to being exposed to chemicals like atrazine, they're still exposed to chemicals like chlorpicrin, which was originally developed as a nerve gas in World War II. We're still using chemicals like 2,4-D, which is a component of Agent Orange. One of the things that struck me, I started digging more and more into this environmental justice issue. I'm a little boy who likes frogs, but now I'm starting to realize that there are other implications for the work that I do out of curiosity. I started thinking about California, and I learned a few things that you may or may not know. Turns out we're the 5th richest country in the world. If California were its own country, we'd have the 5th largest economy in the world. We'd be the 5th richest country in the world. Not because of tech, because of agriculture. 1 in 10 jobs are in agriculture. 30% of the land is in agriculture. We produce 350 agricultural products. And this one blew me away. 50% of the U.S.'s food comes from California. Half of the United States' fruits, vegetables, nuts, berries, and dairy come from California. Half of the U.S. counts on us for food. We use more pesticides than any other state, and 90% of the workers are Hispanic. Now, here's the part that really blew me away. In red now are the top 10 counties for agriculture in California. That's about 30%. So these are the counties that make us the 5th largest economy in the world. Where do you think the 30 poorest towns are in California? So the people who make us the 5th largest economy in the world, the people who grow that food, are the unintended targets of chemicals that we know are associated with adverse health outcomes. And that kind of moved me in a different way. The other half of the equation is, does atrazine turn on aromatase? I asked you to remember it. It causes you to produce estrogen. And let's not talk about any more egg production. You're not going to get eggs in your testes, don't worry. But aromatase is important in both breast cancer, mammary cancer, and prostate cancer. The top cancers in men and women behind lung cancer, depending on the demographic. With regards to prostate cancer, the company published a paper showing that their plant in San Gabriel, Louisiana, there's an 8.4-fold increase in prostate cancer in their factory workers. They published this in the National Institutes of Health Journal. They published this, not me. An 8.4-fold increase in prostate cancer in a community that's 80% black, African American. I'll tell you why I bring that up in a minute. There's also a study showing, with a very significant p-value, that women whose well water is contaminated with atrazine in Kentucky are more likely to develop breast cancer than women who live in the same community but don't drink their well water. Now, that's just correlation, but the company showed that if you take rats, there's control rates of breast cancer, and give those rats atrazine, they develop more breast cancer. It's just a correlation in humans, but we can model that in rats, which we study because they're mammals, like us. What's more is if you take a human cell line, and we've published this, but this is a study that was published by somebody who worked for the company. If you take a human cell line and normalize it to 1 for aromatase production, and you give it atrazine, it starts making aromatase. It starts producing estrogen, just like we showed in fish, just like we showed in amphibians, just like we showed in reptiles, just like we've shown in birds, just like we've shown in lab rats. This is a human cell responding exactly the same way. There's your p-value. I went to visit him. Yoo-hoo, I knocked on the door. Syngenta. Personally, I think it should be spelled with an I instead of a Y, but that's just me. They have I've made the suggestion, but I see they haven't followed up on it. I know my what do you call it, autofill on my phone fills it in for me. They have a pipe that flows right into the Mississippi River. 1.2 million pounds of atrazine flow into the Gulf of Mexico every year. As far as I know, nobody's studying the impact. 1.2 million pounds in a community where much of it looks like this. It's 80% African American, 80% black, low-income community. I bring that up because what you're looking at now, the top 13 cancers that you're going to get in America, in the U.S. In red now, 11 of the 13 are the ones that you're more likely to get if you're black. Being black's a risk factor. What you're looking at now is mortality rates relative to white or Caucasian Americans. If you're black, you're more likely to die from 13 out of 13 cancers. I can show you similar data for Hispanics, and it's corrected, these data, for socioeconomic and for access to health care. Now, is this biology? Racial difference? Could be. Or is it environmental? When the doctor tells you that you're more likely to get breast cancer if your aunt, your sister, your daughter, another female in your family has breast cancer, is that genetics? Or is the doctor just saying, no, you've been exposed to the same crap that your female relatives have been exposed to? If you're a minority, you're more likely to live in and more likely to work in areas that are contaminated where you'll be exposed to chemicals that we know are associated with adverse health outcomes. My other big issue in the way we do business right now is, HeLa aside, every one of those cancers up there, we use human cell lines to try to find the cure. None of those cell lines that are characterized and published are from minority men or women. So when we find the cure, it may be irrelevant to the people who are more likely to get and more likely to die. And I'm all about combing for the cure, but how about combing for the cause? They invited me to give a talk. First, I didn't know why they invited me to be the keynote. They don't give me no money for my research. But I went, and I entitled my talk, An Ounce of Prevention. They didn't invite me back. But I think those are things we need to think about. One of my graduate students, I try not to talk about my graduate students' work because they need to use their work to get jobs and make names for themselves, but one of my graduate students showed that if you take a human breast cancer cell and expose it to atrazine, you can see how the band gets brighter. It'll start making aromatase. Now, the reason that that's bad is because breast cancer is estrogen-dependent. And the reason you get an estrogen-dependent cancer after menopause, when your blood levels of estrogen are lower than they've ever been in your life, is because, one, your lifetime exposure to estrogen and estrogen-like molecules is important. But equally important is your damaged cells, when you get breast cancer, make their own aromatase. I asked you to remember the word. They make their own estrogen, and they stimulate these damaged cells to turn into tumors. You don't have to believe me. You can believe the pharmaceutical companies. Because the number one treatment for breast cancer right now is a chemical that blocks aromatase locally, that decreases estrogen, so that when you get damaged cells, the cancer cells don't turn into a tumor. Now, are you thinking what I'm thinking when I show you that? Are you thinking, gee, what about the number one contaminant of drinking water that does exactly the opposite, that turns on aromatase, that increases estrogen, that induces breast cancer in rats and is associated with breast cancer in humans? In fact, you're probably not the only one who thought about it. Novartis Oncology, this is off their website. Novartis Oncology offers treatments that range for cancers that range from breast cancer, blah, blah, blah. So the same company that gave us 80 million pounds of atrazine in the year 2000 started selling us a chemical that did exactly the opposite. So that when you were taking letrozole to treat your breast cancer, you were drinking a chemical from the same company that did exactly the opposite. And I know Syngenta has a note taker here. They're supposed to be following me around. So I want to make clear what I'm saying. Write this down. This is factual. You can go look it up. In the year 2000, Syngenta discovered that atrazine turned on aromatase. Later in that same year, in July, Novartis broke off from Syngenta and stopped selling atrazine, and Syngenta sold it. And in that same year, they started selling letrozole to block aromatase. I'm just saying it was just an interesting coincidence. I'm not saying that people sat around a table and said, hey, let's invent a drug that does the opposite of our number one selling pesticide and then break off a different company so we aren't both selling the same drug. I'm saying it was just a coincidence. Make sure you keep that in your notes. Just a coincidence. I wrote a paper, though. I called it The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. Pissed them off. They've written letters to my dean, and I told them, I said, you're writing to the wrong person. You need to write to my daddy if you want something to be done. And in the letter, they said something like that I had defamed the company, and they said something like, we don't put our company in that community because it's black. We put our company in that community because it's a low-income community, and that's where black people like to live. And I was like, what? How is that better than what I said? Oh, my. So I think what's happened is, I think that my interest in this aquatic organism has told me a great deal about this aquatic organism. Was not what I tried to set out to do. I would argue, for example, that frogs and humans, we both need testosterone in early development. We both need estrogen. We both need thyroid hormone. Exactly the same hormones, made exactly the same way, that work in exactly the same way. So I would argue that one of my tadpoles, trapped in a contaminated pond or a contaminated aquaria, is no different than a human fetus trapped in a contaminated amniotic fluid. No difference, very little difference at all. Studies have shown, and we use rats to predict what's going to happen in humans, that atrazine causes prostate and mammary cancer, or breast cancer in rats. These aren't my studies. I don't work on rats. Somebody else published this. Other studies have shown that atrazine causes immune failure in rats. I showed the same thing in tadpoles, but this isn't my study. Other people published this. Atrazine causes neural damage when pregnant mothers are exposed. And in fact, I'm not only one of the few academics who brags about what I didn't do. What I'm about to show you now moved me much more than any of my own studies. I didn't do these studies. An EPA laboratory did a study where they showed that atrazine causes abortion in rats. It creates a hormone imbalance so that the rats can't maintain the pregnancy. An EPA laboratory published a peer-reviewed study that showed that. A second EPA laboratory showed that if those rats that don't abort, the sons are born with prostate disease. The sons are born with the prostate of an old man. A third EPA laboratory showed that if those rats that don't abort, the daughters are born with impaired mammary development. This is from their paper. And when those rats grow up, their offspring have retarded growth and development because they can't make enough milk to feed their babies. This is what moved me. Because see that rat on the bottom? That rat on the bottom never saw atrazine. That rat on the bottom was affected by atrazine that its grandmother was exposed to. That rat on the bottom never saw atrazine. That rat on the bottom was affected by atrazine that its grandmother was exposed to. So when I think about my little girl, she's 18 now, has her first boyfriend. When I think about my grandchildren, your grandchildren, could be affected by chemicals that we're using today, it gives me a whole different sense of social responsibility than a little boy who likes frogs, than a tenured professor in the ivory tower. My grandchildren your grandchildren could be affected by chemicals that we're using today. And somebody in the audience always says, just because it happens in a rat doesn't mean it happens in humans. Then why the hell do we study rats? And if it happens in a rat, and there's even the chance that it can happen in us, why would we keep applying this stuff? Our grandchildren. The studies are already here. This is a colleague, Paul Winchester. I met him through the atrazine thing. And what he's showing here in the gray is he's showing atrazine levels, peak atrazine levels. And what he's showing is that if you get pregnant during peak atrazine contamination, you're more likely to have babies with birth defects. And what I want to point out, you just heard a lot of information about the 83% contaminated water here in Hawaii, but none of it's above the government level. I want you to look at the level of atrazine that's associated with these birth defects. 1.2. That's almost 3 times lower than the EPA safety standard. It's associated with birth defects. I notice there's some children in the audience. I'm very happy to see kids in the audience. I may ask you to maybe cover your eyes for the next few slides. Here's a paper that just came out showing you the types of birth defects that we're talking about. It reports that maternal exposure to surface water atrazine is associated with fetal gastroesthesis. I can't even pronounce it, but I can show you what it looks like. Here's another paper, and these aren't my papers. These papers are published by the National Institutes of Health, the Center for Disease Control. These are peer-reviewed, published papers. Here's another one that shows that atrazine is associated with a birth defect called choanal atresia. It's where the nasal and oral cavity don't fuse, so the baby ends up with a hole in its face. And here's another paper that just came out a little less than 2 years ago that shows that atrazine is associated with male genital malformations. If you get pregnant with a male child, with a son, atrazine is associated with hypospadias, where the urethra doesn't go all the way through the penis. Atrazine is associated with cryptorchidism, where the testicles don't descend into the scrotum. So you end up with an empty scrotum, which if not treated, you end up with testicular cancer. And atrazine is associated with micropenis, where the penis simply doesn't develop or grow. Here's what's interesting about this from a scientific perspective. Guess what hormone is necessary for the male genitalia to develop? Testosterone. And guess what chemical we know, this is just a correlation, but what chemical is it that we know decreases testosterone production? In every vertebrate, in fish and amphibians and reptiles and birds and rats, atrazine decreases testosterone, and if you get pregnant with a male child, the genitals are more likely to not develop properly. So this is not about just being exposed as an adult. I like to use this. This is a guy who doesn't like me. He's actually on Syngenta's side. We're not just being poisoned as an adult, but what you need to understand is that everything you eat and everything you drink and everything you breathe in and everything you absorb across your skin when you're pregnant has the capacity to reach your developing child. In fact, studies show, not mine, somebody else's, that we're exposed to up to 300 chemicals before we leave the womb. Before we leave the womb, we will be exposed, your children will be exposed to over 300 synthetic chemicals that do not exist in nature. And most of them, we have no idea what they do to the developing fetus. Even after the baby's born, everything you drink, everything you eat, everything you absorb, everything you breathe in has the capacity to end up in the breast milk, which is nourishing your child. And even after that, a little bit of poison to a baby or to an adult is a lot of poison to a developing baby. Most of these chemicals, we have no idea how they affect IQ, how they affect development, reproduction, the likelihood of developing cancers. We live in a country where supposedly we're all created equal, but what happens after that is anybody's guess. I'd like to read to you now something that the EPA said in a New Yorker article that was published about my work fairly recently. This is a quote from an EPA spokesperson. Quote, "A monetary value is assigned to disease, impairments, and shortened lives and weighed against the benefits of keeping a chemical in use. So a price tag is put on my head, on your head, on your children's heads, on your grandchildren's heads." The EPA said this. And that price tag is being weighed against how much money we'll make on that chemical. That worries me because I know that the price tag on my little girl's head is different than the price tag on some other people's heads. I know that the price tag on Hano, who I just met here on Molokai, that the price tag on his little girl's head is different from the price tag on the heads of people who make these chemicals and who are spraying them. And that concerns me. That concerns me so much that a few years ago, I crossed the line. I crossed the line in the sense that I was taught, don't be an advocate, let the science speak for itself. And I've received some criticism from the universities because I say, I don't want my kids, knowing what I know, I don't want my kids to be exposed to this stuff. And so I should bring the same information to you because your kids and your grandkids shouldn't be exposed either. I asked myself, how can a science speak for itself when the EPA says that the ultimate decision, they said this about my work in 2006, the ultimate decision, they said, is much bigger than science. It weighs in public opinion. And I thought about my mom and my highfalutin publications in PNAS and Nature, and I thought, how is my mom supposed to have an opinion when I'm publishing work in places that she doesn't have access to? That's what you get tenure for. How's my mom supposed to tell the EPA what to do if I let the science speak for itself and she doesn't understand that language that we use? And that moved me in a very different way. I was moved in a very different way because how's the EPA supposed to make a decision, even if it's based on public opinion, when they hire guys like this to be on the EPA panel? This guy, Klaus Werner, was on the EPA panel to review atrazine and got a paycheck from Syngenta and came back and presented to that panel again. How's the FDA supposed to, and I love Obama, I voted for him twice. Not in the same election, but twice. But when he come home next time, can you ask him what he was thinking? Putting a VP from Monsanto in charge of the FDA, asking the chemical company to be in charge of the agency that's supposed to regulate the chemical company. That's a problem. Absolutely. That's a problem when, you've probably seen this figure, 90% of our seeds in blue are owned by 6 chemical companies. And I want to make very clear here, here's my problem with GMO. There's a conflict of interest when the people who own the seeds are modifying the seeds so that you have to use more chemical. I don't want to hear any more of this crap about feeding the world. The #1 crop in California, or the #1 crop in the U.S., and I don't mean crap the world needs to be fed, but the crap from these companies. The #1 crop in the United States is corn. Atrazine increases corn yield by as little as 1.2%, and we eat less than 2% of the corn we grow directly. 20% of the world dies of starvation. We're designing corn, a crop that we eat less than 2% of, to be dependent on a pesticide, so we have to use more pesticide. That's my problem with GMO. How can I let the science speak for itself when Syngenta says that they assume no obligation to update forward-looking statements to reflect actual results? One, pardon my language, children, but who says shit like that? And two, what is it supposed to mean for me and my children and my grandchildren when this is on their website? You may have heard recently that they lost a lawsuit, $105 million. And despite what you might read on their websites, I got no money for serving as an expert witness in this case. I helped with the science. I read papers. I provided papers. I didn't take a dime. Guy took me out to lunch once. But they like to say, I'm a high-paid expert witness, and I'm working with the issue here in court, and I'm not taking a dime. Some of this money went to Hawaii, and this lawsuit was to get atrazine out of the drinking water. But the most significant thing about settling the lawsuit for me is they had a whole bunch of notes that they wrote that they thought were privileged and confidential attorney-client privilege, and the courts made them public. The courts made these notes public that are now allowing me to talk about something that I've been faced with for 15 years, but I couldn't talk about it, and now I can, thanks to Syngenta. I want you to look at some of the things in their own handwriting. Their science strategy for atrazine. Look what their #1 goal was. Discredit Hayes. They wrote that down. It doesn't say test his hypothesis. It doesn't say review his data. It doesn't say reanalyze his data. It says discredit Hayes. They spent millions of dollars trying to dig a hole in my science. And then they couldn't. They went after me personally. They went after the press. This is a news report. How Syngenta investigated the press. They paid people like your boy, John Entine. He's paid by Syngenta. I've seen copies of the bills and the checks. He's a paid third-party pundit. His name is on a list that Syngenta had to turn over to the courts that said sympathetic paid third parties. His name's on that list. Those aren't his thoughts or his words. Here's a list that they wrote at one of their meetings of things they were going to do to me. At the top, they wrote, more risky. Have his work audited. I'm reading to you their handwriting. Ask journals to retract. Investigate students. They scratched that one off. I don't know if that means they did it or they couldn't find anything. Investigate funding. Investigate wife, family background, school history. And look at this one at the bottom. It says, set a trap. It's like something out of some kid's comic book, right? And here's the one that really blows, and then I'm going to move on. Blog psychology, more risky. Psychological profile. My understanding is they paid somebody tens of thousands of dollars to figure out if I was crazy or not. Right? 4 responses to that. 1, you ain't got to pay nobody $10,000 to find out if Tyrone crazy. You can just ask anybody who know me, and they'll tell you, oh, yeah, the brother's crazy. That's number 1. Number 2, what's your outcome? He comes back and he says, no, he ain't crazy. He just pissed off a black guy. Because some people get those two things confused. We're an emotional people. Third possibility is he come back and he say, oh, yeah, he's crazy, and he's whipping your ass. The fourth thing, my fourth response is, who's crazy? They wrote this down. They sat in a room and wrote down, set a trap, buy his name on the internet. And the first page of the Secret Evil Spy Handbook is you don't write it down. And if you do, you shred it, you burn it, you eat it, you self-destruct it. You don't write it down, turn it over to a judge, and say, oh, wait, that was supposed to be secret. This is one of my favorites, too. It says, if TH, that's me, if TH involved in scandal and virals will drop him. They spent millions trying to involve me in a scandal. Did y'all drop me? They should have asked. I know I got a few skeletons somewhere, and they couldn't find them. Here's what's more disturbing to me as I close. The EPA wrote this in an email to me. It's now been unsealed, and it's now part of public documents. The EPA wrote, quote, it is unfortunate but not uncommon for registrants to sit on data that may be considered adverse to the public's perception of their products. Science can be manipulated to serve certain agendas. All you can do is practice suspended disbelief. So this is the agency that's supposed to be regulating the chemical company saying, yeah, we know they lie. We know they sit on data. We just practice suspended belief. Let me give you an example of some of the things they're sitting on. I just discovered this in their notes about a week ago. This is from 2006 from one of their meetings. They wrote, TB, privileged and confidential, you see it at the top? TBA may be a bit more potent than atrazine. Lower doses cause same effects. A couple important things are here. One is they're admitting that they know atrazine does things, right? They say maybe a bit more potent than atrazine. Lower doses cause same effects. They're admitting that they know. Here's what else they wrote. They wrote that TBA increase in mammary tumors, increase in testicular tumors, decrease in body weight gain, decrease in survival. Bit more potent than atrazine. You know what TBA is? Terbutaline. You know what that is? That's the chemical they replaced atrazine with in Europe when they banned it in Europe. So they knew. They wrote this down in their meeting. They knew that atrazine did things. They knew that this chemical was worse. That's what they're using in Europe now. So in light of this, don't be an advocate, let the science speak for itself. I've had to choose what to do. I had to go against the advice of my PhD professor, Paul Licht. And I love the guy. It's a different time back then, I think. And now I follow the following philosophy. Those who have the privilege to know have the duty to act. This guy said that, by the way. Sometimes you learn things in unexpected places. I didn't start out privileged. My father made $9,000 a year. But I've gone to Harvard on a partially public scholarship. I have a debt to pay back. And I'm doing my best to do that. What do you do when the EPA says it's waiting on your public opinion to make a decision? What do you do when, Teresa, I forgot your last name, just sent me these data. What do you do when hundreds of pounds of this stuff is being used in your home now. This is data from Kaua. You can see who's using the atrazine. I say, one, you have to keep doing what you're doing. And a little abstractly, you have to beware of false prophets. This is not necessarily the future of agriculture. There are alternatives. There are different ways that we can go. If we really want to feed the world, we can do some very different things and start to do that. The second thing is, in addition to bewaring false prophets, beware of false prophets. Because the money that they're making and the GMO that they're developing and the pesticides that they're using, that the EPA is weighing against the cost on your health, that money does not come back to this island. That money does not go back to you. That money does not come back to me. Legislation, litigation, regulation. You got to push all three until one of them, or a combination, is successful. Finally, I'm going to end with a quote that you also might think is from an unexpected place, but it's from somebody that I've grown to respect and learned a lot from. It's time for us as a people to start making some changes. Let's change the way we eat. Let's change the way we live. Let's change the way we treat each other. You see, the old way wasn't working, so it's up to us to do what we got to do to survive. This guy said that. Thank you. I want to thank you. This is my last time sharing on the islands this trip. I'll come back anytime you ask. Next time I'll take at least a day off. But it's been great. I have not been bored. I have not been hungry. And I have not felt, anytime I go away from my wife, my son's back home from college now, from my daughter, from my lab, I have to make sure that I'm doing something that's worth me being away. And this was definitely worth it, and I thank you very much again for your time.